I would give a credit to the landscaper who brought me some older boxwood samples. But he brought me only like a twig. Okay. So when we saw the twig and we have seen those symptoms very similar produced by another root rot pathogen, we thought it was Phytophthora root rot. But unfortunately, we were unable to detect any Phytophthora through culturing or through immunostrip detection. We couldn't find it. We worked on the roots. We tried different methods to isolate the pathogen, and it was not there. So we closed the case saying that the symptoms look like Phytophthora root rot, but we didn't detect it. So it was kind of like a partially solved case. So the landscaper didn't give up. So after about 2 months, he called us again and said, did you find anything on it? So it kind of reignited our research then. So I was surprised that even though we couldn't find it, it was spreading in the landscape. And it was a big church, not like a small landscape. It was a huge, and I'll show you a picture of that too. It was a big landscape. So it was not kind of like neglected or anything like that. It was well groomed and it was well taken care of. So that's when we went back to the site, and then we started finding out more and more. So that's when we first found out that this is a Calthotricum, which causes anthracnose kind of symptoms on other ornamentals. Okay. One of the Calthotricum anthracnose is very common. It's on strawberries. Okay. And there is a bitter rot on apples. Okay. I just wanted to give you an idea of what Calthotricum I'm talking about. So it's known for causing anthracnose. And in the past, it has been shown that boxwood, they have Calthotricum, but these symptoms were never revealed before. It was more kind of like an endophyte that it can stay inside the host without causing those symptoms. But in 2011, we found out that this is actually causing the problem. So based on those 4-year studies, we first published a report in 2015, and we had it from North Louisiana, New York, Virginia, North Carolina, South Carolina. So that was the first official report then. Okay. But then in 2018, Texas reported same thing from Houston area. So it is also in Texas. At this point, we know we have found it on Japanese boxwood, Korean, English, baby gem, and other hybrids. Now, when I talk about other hybrids, there has been only few cases. So they are significant, but at this point, they are kind of like random in some of the states. So we are not finding them in every state that has this disease. Like I said before, the symptoms resemble 3 major diseases that you will find on boxwoods. One is Volutella blight. And I think if you are in the nursery business or in the landscape business and you deal with boxwoods, you have seen all these diseases before. So you have Volutella blight that causes similar kind of symptoms. Macrophthalma blight, which is kind of similar, but not exactly. You won't be able, you can easily distinguish between Macrophthalma blight and Boxwood dieback. And obviously Phytophthora root rot it's kind of, I'll show you a comparison of those two, Phytophthora root rot and Calthotricum theobromicola, Boxwood dieback, that they look very similar. And that's when I started thinking about that there could be a problem. Like the first case that came to us, we thought that it was Phytophthora root rot because based on the symptoms, okay? In diagnostics, sometimes we do that. Sometimes we say that there is a disease, but we can't detect the pathogen for any unforeseen reasons, okay? So right now it's present in nurseries and landscapes. So we have received samples, not just from the landscape, from the nurseries who specialize in boxwoods, who have some boxwoods, and these are from wholesalers and retailers. So it's kind of everywhere right now. First, we knew when we first dealt with this disease, we thought it was only a foliar pathogen. Foliar in the sense that it's not attacking the root system of the affected boxwoods. But I would say last 2019, we found something very interesting. Okay, and I'll show you that too. So just to give you an idea that it is been spreading, and we are finding new aspects of this disease which we didn't know about that. Okay. So this is the original picture that was taken in 2011, and you can see the random dieback in the canopy. If you look at the size of that boxwood, that's a good size boxwood, and the landscaper, they didn't give us the accurate age of those boxwoods, but he told us that these are very old boxwood that were kept very small in a hedge form. Okay, but you can kind of like make an assumption that these boxwoods are more than 10 years old, looking at the stem or the crown of those boxwoods. So when we talk about these kind of symptoms, we either find a pathogen where the disease is causing, or we find the pathogen in the root system because the roots are unable to pick up the nutrients and water, and then that's causing the dieback. Okay, but there are other foliar pathogens like a volatile blight that will produce similar kind of symptoms. But then we looked at the root system, and those root systems were perfectly fine. Okay, no problem at all. The roots were healthy. They were feeder roots. And when I had to dig these up, the affected boxwoods, I had to use a shovel. And it took me some work to dig them up. It was not an easy just pull right out of the ground. So I had to do some hard work you need to dig them out. Okay, and you can see the roots are perfectly fine. But at the same time, with that kind of root system, you're seeing the dieback in the canopy. So it was kind of puzzling at the moment that we thought that these are because the symptoms are caused because of the root systems, but there was nothing wrong. And then we also were thinking that if we didn't find the pathogen, maybe the roots are not, they're in a compacted zone, or maybe they're waterlogged. But you can see how loose the roots are. Okay, there was no compaction, there was no root injury to it. So that was again puzzling, because the symptoms again are most probably caused by the root rot system, because the roots were rotting. But then we worked, like I described before, that we worked on it for about a month or so. We couldn't find the Phytophthora or any other root rot pathogen. So we started digging more and more. And so one of my colleagues who passed away in 2012, he suggested, why don't I look upper in the canopy? And he was actually my committee chair when I was hired at LSU, and then he became my mentor. So he suggested that why don't I look up in the canopy? And I followed his instructions and started looking in the canopy. And this is what we found. Okay, once you scrape the bark off with a sharp blade, you can see there is some dieback in the stems too. And the picture on my left is up in the canopy. Okay. And the picture on the right is something new that we are seeing. Before we thought it's only up in the upper canopy. Okay, the crown is not affected by it. But this picture, the picture on my right hand side, that was taken in 2019, where we found that it's not only up in the upper canopy. This is the picture here. You can see it's in the crown region. Okay, so this is something new, that is not only in the black discoloration of the tissue is not only up in the upper canopy, but it can also happen in the crown region of those boxwoods. Now, if you look on the picture on the right, you can see, this is not actually a good picture, but I just wanted to compare to show you something. So you can see some of those fungal structures here. Okay, those black dots. When you look close, you can see actually the CT coming out of, that's one of the fungal morphology we talk about. Okay, but you can see the pathogen is present here. And again, same thing, when you remove the bark, you can see the discoloration. You can see it's extending both ways, upwards and downwards. So in this case, you don't know where the infection started. Before 2019, we were just thinking that if you are pruning the tools, if you're pruning and and trimming those boxwoods, that could be a source of your transmission from infected to healthy boxwoods. But 2019, it changed. We found it that it's not only in the upper portion of the canopy, it is also present in the crown region. And you can see the thickness of that stem is quite thick. When you have a section of it, you can see how that same discoloration is extending towards the center of the affected stems. Okay, and we were able to isolate. Generally, what happens is when you have a dead tissue like this, you won't be able to get any fungus out of. But in our when we first started working with it, we were able to isolate Caltotricum from these sections as well. Okay, we just plated some discs on artificial media, surface sterilized those discs, and then put it on artificial media, and we were able to consistently isolate Caltotricum from those affected stems. Now, it doesn't stop there anymore. Okay, these were pictures. Now, this picture on the right-hand side, where you see a zoomed photo of a leaf that has a leaf spot. I would say, I'm going to call it leaf spot. It has a leaf spot on it, and in the center of that leaf spot, you can see the same fungal structures. We call them servolus. Okay, these are servolus. So this is where the fungus is present, and you can see some of those setae. They are black hair-like structure. They stick out. So those are two diagnostic features of this fungus of Caltotricum: servolus and then your black setae. Okay. Now, this picture was taken in 2014. Okay. At that time, I didn't think about that the same fungus can cause the leaf spot symptoms too. But then one of my colleagues at Purdue University, somehow he received a sample of affected boxwood from one of our major nursery here in Louisiana. And I was told that their chemical rep sent those samples to Purdue University for some reason. And they contacted me and said, well, we see something very similar that again looks like Caltotricum. Have you seen this before? So because I have seen those symptoms before, so I compared those two and it looked very similar. So I received those samples from John at Purdue University, and we confirmed that it's the same fungus. So this is something new for the industry, that it's not only on the twigs, it's not only on the crown or the stem, it can also be present on the leaves. And on the picture on the right, you can see there is a central spot there, and then there are spots on the edges of the leaves. And if you pay close attention to this newer twig, okay, you can see there is also another spot on that twig. Okay, and you can see it's present on the edges too. So this is something new that we have to address in coming in the research that we're going to do, is that if it causes these symptoms and those leaves are dropped, then that could also be a source of inoculum. Okay, so previously, again, going back, we were thinking that if you're trimming or hedging your boxwoods, that could be the source of transmission. Now, in this case, if you can see how many servolus are being produced in one spot, and they're going to produce, and you can see the picture on the right. If you look at the top, those are actually the cnidia, okay, that are formed, that are produced out of those servolus. So if you are using overhead irrigation or something like that, which will, or even the water splashed from rain will spread those spores to the nearby healthy plants. Like I said, it's not just in the landscapes, it is present in the nurseries, it is present in the retail stores, it is present in the wholesale boxwood business. And this picture is from Alabama. You can see these are liners, okay? And you can see what percentage of these liners are affected. So if even if you take out those liners that are showing symptoms, okay, you can't tell the rest of the liners won't have the disease. So if you are just trying to get those healthy looking liners, you can easily transmit the disease from one point of region to where those nursery liners are going. So it's very, very important that we have to, when we're talking about Boxwood dieback management, we have to do it from all aspects, from all different angles. We can't just worry about landscape anymore. This is another plant, like I'll talk about the wholesale nursery. This is a baby gem boxwood, and the picture on the left, you can see the center of that plant is kind of off colored, okay, compared to the rest of the plant. So this is kind of off colored. So when you go inside the canopy at the same twig, that is with the off colored foliage, when you do scrape the bark off, you will see the same discoloration of the tissue, discoloration of the stem tissue. Okay, so at this point, if I'm just looking and I'm not paying attention to what symptoms, these are kind of early disease development symptoms, early symptoms. So if I'm not careful with these kind of symptoms, and I sell this plant, obviously I'm going to have some issues because it's already being introduced into the landscape now. And this is an interesting case. Many of you might know Alan Owings. He retired. He is with now nursery business. He was one of our ornamental specialists. And this picture, he brought me these plants, and he got mad at me. These were baby gems planted in the landscape only like 6 months when they started showing symptoms. And he said, baby gem is kind of a newer cultivar, so how this disease can go into the new cultivar. So this was again something new, that these new cultivars are so much affected with the disease, and you can see how widespread this is in the smaller plants as well. Now, let me tell you one thing. At this point, we don't know if from any disease point, pathogen point, we know smaller the plants or younger the plants, they are more susceptible to disease problems. But in this case, we have seen the older plants showing the same symptoms, the younger plants showing the same symptoms, the liners showing the same symptoms. So at this point, we don't really know if that the age of the plant is going to have anything in terms of disease development. Okay. At this point, all we know is that it can happen on any age plants or any size plants. Now, this is again in 2019. That's when we changed our opinion about this disease. We were thinking that it is mostly a foliar disease. So this is a landscape, and it's a big house in one of our cities. And I would say if I had counted those boxwoods, there were about like 10,000 boxwoods. This is just one section, one side of the house. And this is a big house, and there were boxwoods everywhere. You can see from this section. And you can see the type of disease present, the disease pressure present in that landscape. Every single one of them had the same symptoms, okay? So when we looked at the foliage, so in the inset, you're seeing that foliage, and when you see at the foliage, you can see the same acervulus present, and it's producing the cnidia, the spores. Now, interesting thing about this cnidia is that they're sticky in nature, okay? So that's when we thought that when you are pruning or hedging or trimming, maybe if there is cnidia present, maybe what's happening is that when you're trimming, those cnidia are getting stuck on the trimming tools, any kind of, like if you're using hand tools or gas powered tools or anything like that to hedge them. So this is what we are saying, that the cnidia, they get on the tools, and when you go from one section to the other section, because you are causing an injury when you're trimming, okay? So that's when the cnidia has a potential to go inside the system and cause the disease. So a little bit more new thing that we found. So this is the same property, but a different section. Now you can see in the middle, you have two boxwoods that were removed, okay? That were previously removed because they were totally dead. Now the two boxwoods on the two other sides, they were healthy looking, very few symptoms, very small amount of symptoms in the upper canopy, okay? Same here and same here. But when you look at the plant from a distance, you won't be able to tell that if they have the same disease. Now, these were removed at least 6 to 9 months before, okay? So, and these were starting to show symptoms. So I said, well, why don't I look in the crown, because we now know it can be present in the crown. So in the inset, you can see the same kind of discoloration that's happening on this healthy looking plant, okay? Now, if you look at the other pictures that I've shown you before, where we're seeing that black discoloration, you see a large amount of symptoms, the dieback in the canopy. So with that kind of discoloration here in the crown, you will see this whole canopy died back, okay? But in this case, that was not true, okay? So what we decided that why don't we see if the disease is not just in the foliage, what about the roots? What about the soil around it? So fortunately, these two, I would say, stumps were there. We dug them out and we brought them back. We cleaned them up, everything. We tried to make sure that we are not contaminating our stalks or anything like that. So we were really careful that we are not trying to transfer the disease which is not present in those stalks. But it was kind of a surprising discovery here in this case. So when we saw this is the tissue from like the crown and the root, where the roots are present. So you can see the same acervuli, okay, with CT. CT are those black hair-like structures, okay, and then acervulus. You can see it in the inside that they are present in that section too. So this is from this section over here, right there, okay. So this is not from the upper canopy. The picture is from the stalk. So the fungus was present in there. So we thought maybe it's just present because the tissue died a long time ago. The upper canopy was taken. So maybe it's just sporulating in order to survive. So we thought that it's just like acting as a saprophyte, which is kind of like in a survival mode. So we thought maybe we need to do some more digging into it. So that's when we did this assay, okay? You can see we have 4 plates here. You got upper stem, crown, roots at 0 to 2 inches, and roots at 2 to 4 inches, okay? It was surprising to see that the fungus was present in the upper stem, okay? And then the crown region where I showed you that the area between the roots and the actual stem, and then roots at 0 to 2 inches, and the roots at 2 to 4 inches. So which is very concerning now, okay? Now, when we plant boxwoods, especially here in Louisiana, I have seen that we put boxwoods next to each other, like a hedge. We use it as a hedge. So this is kind of concerning in the sense that if those roots are cross-grafting with each other. Remember I showed you that stem that has transfer section of a stem that had the fungus killed the part of the section towards the center of the stem, and we were able to isolate pathogen from that. So if that's the case, and if the roots are, if the pathogen is present in the roots, so if the roots cross-graft with each other from 2 different plants, because they're so close to each other, and we have seen it, we see it like they cross-graft, they kind of go in between each other. So if there is a chance that they're cross-grafting, or even if coming in contact that has an active pathogen, that it can be transferring the pathogen through the root system, which is again the case here. We don't see the type of the symptoms that we are supposed to see with that kind of that high amount of discoloration in the crown, we don't see that symptom. So this is a good supporting evidence that the disease moved from these older plants to the nearby plants. Okay. Now, scientifically, we have to prove it by doing some assay where we take two different liners, put one liner in the disease media, and then one liner just keep it for living for some times, and then put them together in a same pot and see if the disease transfer from that inoculated to a healthy plant. So that can be done, but that's in the pipeline right now. But now we know that it's not just present in the upper canopy. It is also, you can find the pathogen actively growing and actively living in the root system up to 4 inches. Now, after 4 inches, there were a lot of contamination. We got Fusarium from it. And as you all know, Fusarium is everywhere. In some situations, we call it a pathogenic. In some situations, we just say that it is just a saprophytic working on the dead tissue. Now, when we come back to inoculations, like how long does it take for the symptoms to develop? Now, these were in the greenhouse situation. You can see 6 weeks after inoculation, you start seeing the off color. Now, the two plants on the left are inoculated. The 2 plants on the right are controlled. But even in the control, we did the same kind of wounding to the stem. Okay. So when we our inoculation method was that we remove about 1 inch of the bark, okay, like in a rectangle shape section. And then we have the same size of the culture, the pathogen, and we put that together and we wrap it with the parafilm. Okay. So you can see here that you're seeing the symptoms, the off-color foliage or the dieback, and at the same time, the control that didn't receive any pathogen stayed fine. There was nothing wrong with it. Okay. But this was after 6 weeks. And after 6 weeks, we were able to see the black discoloration. Again, this is your inoculated plant, and this is your control plant. You don't see any black discoloration here, even after wounding the tissue. But here you see where we inoculated it, it's going up and it's producing those symptoms. 11 weeks after inoculation, you see the clear symptoms. Now, the 5 plants on this side, these are Japanese boxwoods, okay? So the 5 plants on this side, they were inoculated. 5 plants on this side, they were non-inoculated. It was not just 5 plants each. I had about 15 plants each that were inoculated and non-inoculated. So the story in this case started that we inoculated the plants, we put them in the greenhouse, we tried to create high humidity kind of 20 to 30 degrees Celsius temperature, and high humidity in terms of 80 to 85% relative high humidity in the greenhouse. Being a pathologist, it's always kind of like you're anxious to see the symptoms. Okay. In this case, I didn't see the symptoms within like 2 to 3 weeks. So I got kind of little upset that why it's not producing the symptoms. We re-isolated everything. So I told my research associate, I said, well, why don't you just take care of them? Okay, just keep watering them, see what happens. So that's when he came back to me after like those 6 weeks. He said, oh, what should I do with those boxwoods? And I completely forgot about them, to be honest, that we have some boxwood for a project going on. So I asked him, I said, what boxwood? He said, oh, the boxwoods you asked me to take care of. I'm like, what's wrong with them? He said, they're dying. And you won't believe, I was like jumping off my chair and like, really, they're dying? Do you see them die? So that's when we came out, and that's when we start seeing the symptoms. So long story short, it will take time for the disease to develop. Okay. Even on the smaller boxwoods, now we know that takes up to about 60 days for the symptoms to be produced. Remember, we talk about the accurate identification. Okay. Remember, we talk about why we might have misdiagnose this disease. So if you have to guess which one of them is a Boxwood Dieback. Now, in this case, I know because I showed you the bottom picture before, so you have an idea which one is the Boxwood Dieback. But if I just give you these two pictures, you won't be able to tell which one is Boxwood Dieback and which one is Phytophthora root rot, because the symptoms just mimic each other, okay? So in this case, the top one is your Phytophthora root rot and the bottom one is your Boxwood Dieback. But you can see the similarity in the symptoms, okay? Very, very similar. And in some cases where we have now detected Boxwood Dieback, those plants were treated as Phytophthora root rot. Okay. Now, many of you know Phytophthora is not a true fungus. It is a fungal-like microorganism. It is an oomycete, and it affects the roots and the crown of the affected boxwoods. And here's something more about this is also Phytophthora root rot. In this case, you can see how the roots are totally dead. Okay. Then you can also see the discoloration of the crown region, the sloughing of the bark at the crown region. But this discoloration is totally different from what you saw back in those pictures from Boxwood Dieback. That Boxwood Dieback causes like a bright black discoloration. It's like bright, it's living. In this case, this is like just a necrotic tissue. And if you look at some of the roots of those plants that are affected with your Phytophthora root rot, you will see these lesions, okay? The picture on my right is showing you the lesions, the necrotic lesions, reddish brown lesions on the feeder roots. And also you can see the mycelium that is present from Phytophthora, okay? So these are the symptoms that you will see in Phytophthora root rot, not in the Boxwood Dieback. Now, remember the picture I showed you where the two plants were dead in the center of those two other plants, or in between those two plants? When we dug those out, the roots didn't rot like this case. The roots were still intact, okay? So we were able to dug those roots, and we had to use a shovel to dug them out. It was not easy to pull them out. So the roots were still intact. So that's another difference between Phytophthora root rot. But in this case, if you have boxwood affected with Phytophthora root rot, you may not need a shovel to pull them out of the ground. You should be able to just pull them out. So let's talk about current distribution in the US now. The states that are in bright yellow, highlighted in bright yellow, they have been confirmed positive for Boxwood Dieback. The states that are in bright orange, we have personal communication from these states that they have confirmed the disease or the presence of the symptoms in the boxwood landscape, but they haven't confirmed it officially. When we say confirmed it officially, that they have to go through all different kind of like postulate to establish the relationship between the isolated pathogen and the symptoms. So, but we know that Tennessee is working on this disease to confirm it officially. So there might be another plant disease node that's coming out in near future. So as you can see, it's spreading, okay? Now we know we have it in Arkansas and Mississippi, but again, there are no official reports, but from my colleagues in those states, we were able to find out that they have seen those symptoms, but for some reasons they haven't reported them yet. But you can see how it's kind of like spreading. And one of the reports that came out of California, the symptoms were very similar, but somehow the sample got lost somewhere. So I'm suspecting that it's not just restricted to this part of United States. I think it's present in other states too. It's just a matter of finding it and confirming it. And I believe because it takes too long for the symptoms to develop, some pathologists don't have enough time to work on those symptomology. But this is the confirmed distribution right now that we have here in the US. If you ask me about if the disease is present in Europe, I was in Czech Republic in 2018 in Brno, but I went to Prague on a weekend and I see there are boxwoods everywhere and I can see those symptoms on those boxwoods. Okay. But I don't know what was the main reason they don't want to report the disease or they don't want to work with this disease. But I did a presentation there as well, and I showed them what those symptoms look like and how they can isolate the pathogen and the whole 9 yards about that disease. So that was from, I would say, 2011 to 2018. Now, going back to 2018, I got a student, graduate student, and I would say that graduate student, I never had a graduate student like her before, okay? She's already offered two PhD positions, one at Georgia and one at Clemson, and she did a fantastic job, okay? I'm really impressed with her, and she actually got 2 awards from the department as well. One is very prestigious award. She is like a Student of the Year award. So we had 3 goals for her study. Okay, first one is because we know there is a confusion between the symptoms, so we didn't want to just confirm the disease on visual symptoms. So what we had to do, we had to develop a method that we can do it within a day or so. Okay, because we don't want to wait for 3 months for the symptoms to develop and then isolate the pathogen and then confirm the pathogen by doing 4 different analyses. Okay, so in this case, you can see she developed a method which is a real-time method. It takes only from DNA extraction to running that sample, it takes only like 2 hours to do it. Okay, and we saw that we are able to detect the pathogen. We don't even have to culture it out. We can detect the pathogen right from the plant itself. So when you look here on your right-hand side, L4 to L8, which are these correspond to these sections here. These are the DNA of the pathogen. Okay. So you can see it didn't amplify the healthy plant DNA. It didn't amplify the healthy plant DNA from the greenhouse, but then we had inoculated plants from the landscape and the greenhouse plants. So you can see nursery, landscape, and greenhouse inoculated plants, and it gave us a positive reaction. And so this is the real time. For this, the conventional PCR, it takes about 4 to 5 hours. Then you have to run the gel, and that's another part to it. But in this, we were able to confirm that whatever happened in the conventional PCR, conventional amplification, same thing happened in our real-time PCR. So this is another tool for any nursery out there, or even the landscaper. They want to know that if the disease is present or not, so you will be able to identify it. Same thing, we had isolates from all different states, okay? Alabama, Louisiana, Texas, Missouri, North Carolina, Oklahoma, South Carolina, Virginia. And you can see they amplified from lane 2 to lane 9, okay, right there. It amplified all of them. And then we had some other Calthatricum from other hosts, okay? There were some, one from cashew and 2 from cocoa plant, and it amplified those 2. So it is specific from boxwood, but it is also specific for Calthatricum theobromicula. And same thing happened with our real-time PCR, where it amplified only Calthatricum theobromicula. Now, the second goal was to screen some of the fungicides. So in this case, I wanted to share this information, but I would ask the participant that when you use this information, don't release this yet, because my student is preparing the paper. So we are in the final phase of preparation, submitting that paper. But I want to bring your attention. So in this case, what we did, we selected 9 fungicides, and out of those fungicides, we selected them on the basis that they were previously considered very effective to excellent efficacy against Calthotricum and other host species. So I want to bring your attention to these two. This is your Morel and this is your Positiva. Okay, so these two fungicides, they were able to, so this is our mycelium inhibition. It's in the lab, okay? It's in a small Petri plate where you have different concentration of the fungicide, and then you use those concentration against the mycelium inhibition to see what parts per million is able to kill the pathogen or inhibit its growth. So you can see this is in percentage up to 100%, and you can see these two, okay, Morel and Positiva, even at 1 ppm, okay, the orange is 1 ppm, it was able to control up to 80% of the mycelium, okay. When the concentration went up, you can see very close to 100% of the mycelium was inhibited, okay. So this is very promising. There are others, but these two were kind of like highly effective. But when you look at the others too, so if you look here, that's 60%, that's close to 60%, okay? So they were able to control more than 50%. So in this case, we did the LC lethal concentration 50, where 50% of the mycelium is inhibited as compared to our controls. So this is just to give you an example of what worked and what didn't work. So the top is your control, and then these are the different concentrations. So 0.1, 1 ppm, 5 ppm, 10 ppm, and ppm is parts per million, 25, 50. So the top is least effective, okay? The top fungicide, but at the same time, remember I told you about Morel? It was very effective. Even at 1 ppm or 0.1 ppm, you can see about, I would say about 60%. And then when it comes to 1 ppm, you can see nothing grew out of it. And at 10 ppm, it's just nothing grew out of it. So these are some very promising results that we have produced in the lab, but we now need to take this to the greenhouse study and other things. So this is just a differentiation of what we have done. So these were our 9 fungicides that we tested. Unfortunately, these two fungicides, Camelot and Dithane, didn't do anything. Okay, even at 1,000 parts per million, they didn't do anything. And now we know because these are contact fungicides. Rest of them are your systemic fungicides. So you can see Morel and Positiva that I showed you before at 1 ppm, 78%, which is very good efficacy. And even our thiophanate methyl, which is kind of like our regular landscape fungicide, systemic, broad spectrum, 2636, that was able to give us close to 80% of the mycelium inhibition at 5 parts per million. Okay, and then your orchestra intrinsic, your Azox, the pageant intrinsic, they were effective, but not as compared to the other two. Okay, but still they were effective if we talk about LC50. Okay, so based on that, we kind of put this this is an efficacy chart for, especially with these chemistries. Previously they were considered very highly effective to very good to excellent efficacy against the Calthotricum. And in this case, we found that there were some that were not effective at all, that were considered effective before. So this is going to be published soon, but you have my presentation, so I'm okay to share with that. But just, I would say don't release this information yet. But we are doing good with our fungicide. One thing we noticed that remember we had Morel and Positiva. They did both very good when they were used at 1 ppm, and they gave us about 80% mycelium inhibition. But when it comes to, remember we suspected that the spores are the major transmitter of this disease. When we compared the same, our Morel didn't do as good as our Positiva. Okay, So even at higher concentration, at 25 parts per million, we only got about 60. Whereas with this one, we started getting 90 to 100 from 1 to 25 parts per million. So more research is needed. Why didn't it control the spores compared to when we compared the mycelium? So we don't know yet, but that it could be like another graduate student who would like to work on it and find out why it didn't do it. So right now, this is where we are, and we need to find more, and we need to take this study to greenhouse and then to the field landscape and see if this is true. But we are very optimistic that it's going to behave the same way in the greenhouse and in the landscape situation. When we did the host range, this is kind of a bad, I won't say a bad news, but it's not good news. We did about 11 cultivars that we screened against this pathogen. And unfortunately, all of them are susceptible, okay? We didn't see any kind of resistance in them. That's why I'm saying it's kind of discouraging. And some of these are even new, that's just recently introduced, and some of them haven't even been introduced. So you can see there is a range of days that it takes to develop the symptoms, but it's all over the place, okay? When we talk about Little Missy, it took us longer to produce those symptoms, okay, to reproduce those symptoms. And again, this study is to just see if they're susceptible or not susceptible. The goal was to just see if there is any resistance available, that some cultivars didn't develop any symptoms. So our null hypothesis that all of them produce symptoms, that was correct. So, but again, we want to know that is this true in the field situation, in the landscape situation, or maybe there is more that we can test in future to see if there is any resistance available. But at this point, these were the only one that were available out there, and we could do something with them. So this is my last slide, and these are just the general management practices that are true for any other disease similar to Boxwood Dieback. So I put them together in both like a nursery and landscape perspective. So obviously, if you have disease Boxwood symptoms, now we know that there is a potential for the roots to spread the disease. Now, still we have to prove it, but my gut feeling is, and my professional experience is telling me that it is the case. So cutting out sections that are dead is not going to solve the problem, okay? I have seen landscaper professional doing that. And unfortunately with boxwoods, it doesn't fill out the gap, they fill out the hole that you cut out the dead tissue. So if you cut out the tissue, there is a chance by the time you start seeing those final symptoms of tan colored foliage, the disease is already spreading to the rest of the canopy and rest of the plant. So removing and discarding diseased boxwoods is the first and foremost method to stop it from spreading. And if it's in a nursery, I would say if you can burn those diseased plants, because if you put them in a cull pile, there's a chance that the pathogen can move back into your boxwood blocks. Now we know that foliar transmission is also possible by trimming. So if you are trimming or hedging, make sure your blades are sharp. Make sure you are not just, the idea here is to make a clean cut. If you make a clean cut, if you're not stripping the tissue, then it's much more protected compared to if you are doing it with a dull blade and you're stripping the tissue. That gives a more surface area for the pathogen to work with. If you are a nursery and you are bringing in plants from outside source, I would say that you keep those plants in isolation and just dedicate one of the blocks to like away from your production area and keep those plants there for 6 to 8 months. And I just showed you the slide where it takes, it depend upon what um, cultivar you're working with. So anywhere from 6 to 8 weeks, I would say is the time period when it's going to start showing the symptoms. Don't reuse the potting mix. I know some nurseries do that. If you have a plant that died early in the season and you just take out the plant and you reuse the potting mix, I won't do that because now we know that there could be the pathogen because it's present in the roots too. So you might have that contaminated potting mix. Drip irrigation versus overhead irrigation. If in a nursery, if you can avoid overhead irrigation, that's the best part to fight with this disease. Now we know it can produce spores on the twigs, it can produce spores on the leaves. So any water that comes in contact with it is going to spread those conidia. Uh, I already mentioned prune with sharp tools. It's very important. Pruning tools, disinfestation. You can use any kind of disinfectant. Rubbing alcohol is good. Bleach, if you use bleach, you may want to rinse it with running water because that will corrode your, rust your tools. Ammonia, pine sol, any kind of disinfectant you can use to disinfest pruning tools, maybe when you're going from one property to another or one section of the house to another section, or maybe after every like 2 or 5 or 6 plants. I know it's not very convenient when you are pruning like a whole section, but in order to avoid it from spreading, if you know the disease is prevalent in that area, you may want to do that. I mentioned about those fungicides, but the research is not done on it in the landscape yet. So we need to find out what dose is effective against those against this pest, or how far along we have to apply those fungicides before the disease can come and start infecting those plants. So you can use those fungicides. They are listed for boxwoods. Okay, but again, we need to find out more field data to confirm that effectiveness. Unfortunately, there are no resistant cultivars so far. I didn't put all the previous ones that we know the disease came from or disease was pathogen was isolated, but there are other hybrids that are out there that are known host of this pathogen. So if you have seen the disease and if you have seen the cultivar that is not showing symptoms, I would like to know and maybe get some of those plants and inoculate them in the greenhouse and see if there is any resistance. But I'm not very optimistic about, because we did 11 and all of them are susceptible to this disease. And with that, I would take any questions. And that's my cell number. You can use my cell number too, but I prefer email because I check my email very frequently. As an extension specialist, I'm kind of like stuck to my phone. I try my kids to tell them, don't use that phone too much. And then I'm just checking my email every other minute. But email is the best method to contact me anytime. But you can call me if you want help. And with that, I would like to thank you all for staying along with me. Thank you.
Thank you, Raj. I'm going to let Amanda, if she can, handle any questions. And while she's doing that, I want to just remind everybody, I forgot to maybe specifically say this just to give anybody a final chance. If you are seeking pesticide credits, make sure your camera is on. I'm going to take a few screenshots as a last backup on who is on. And then once questions are complete, I will put the Google form in the chat.
Danny, we don't have any questions in the chat.
That's good.
Danny, if you're trying to talk, you're muted.
Thank you, Stacy. Sorry about that. Yeah, I'm going to do a few screenshots here, so y'all give me just a second. Actually, what I'll do is let me go ahead and put it in the chat for your pesticide license information, the Google form link. I am going to stay on. I just put it up there. I'm going to stay on for a while in case anybody has any issues accessing that. If y'all will please go ahead and go to that form link and submit that information. I plan on going ahead and uploading the pesticide credit information to NCDA around 1 p.m. today. So go ahead and get that done. And again, I'll stay on here if anybody has any issues.
Danny, we do have a couple questions that have come in the chat box.
Okay. If anybody has any issues, just speak up and ask those about the pesticide credits as well. And then, or put them in the chat.
See the link or anything.
Go ahead, Amanda.
Okay, so Tom Woods asks, why do you think the Little Missy cultivar was the most resistant?
It's not resistant. It's just, I think, because when you look at if you go back to that slide, and Denny, I'm okay with, if you want to share that PowerPoint presentation with the participants, I'm okay with that. So when you look at that slide, it took longer for it to develop the symptoms, but it's not resistant. So resistant is that you inoculate it and never develop the symptoms. That will be resistant. I would say maybe it's a little more tolerant compared to the other cultivars, but we need to confirm it. We inoculated it 3 times and we had the same results. So, but when you look at it, all the other cultivars, they are also kind of like, some develop the symptoms after 40 days, some at 90 days. So I'm not going to call it, it's just, I would say the latent period of the disease is longer in Little Missy. So it's not resistant.
Okay, and we have a couple questions just questioning whether you've seen it in any other species, specifically mentioned Pachysandra or Sarcococca, like you would with boxwood blight.
Not yet, not yet. So that's good news, actually. So it's not going to go to closely related Boxaceae families. So, but once we confirm it, the next goal is to get more hybrids in there. And also the closely related, we want to test some of the hollies, ilex species. It's just a matter of getting those plants in. But so far we haven't seen it. And so far, none of the other states have reported it from any other close related Boxaceae family plants, like we have it on Boxwood blight.
All right, great. That is all the questions we have.